It's an honor to introduce to you Professor Kenneth Rock. So he's a full professor at the pathology department of the medical school, University of Massachusetts. Professor Kenneth Rock, I was just talking to him that I ended last night reading his papers. It's very interesting ones. He did astonishing contribution both to innate immunity and adaptive immunity. For example, he showed the relevance of sometimes of catepsins on the activation of NALP3 inflammasome, for instance, very relevant papers. But he's here today to show the other stories that he's very involved with, which is antigen presentation and of antigens by Mhc Class 1 molecules, which of course is very, very relevant for the immune response against the HIV, right? So I wish you all a good talk for you as well, Professor. Bem-vindo e muito obrigado.
Obrigado. Bom dia a todos. Primeiro eu quero dizer obrigado pelo convite. E é um prazer para mim estar aqui. Eu adoro o Brasil. Eu não sou fluente em português e o meu sotaque é ruim, então eu vou falar em inglês. Mas minhas legendas são em português. Então, eu vou mudar e falar em inglês. So my lecture here is going to be one that covers generally the field of antigen presentation and things that my lab has done and other work that has been in the field. So it's really a general landscape of what's going on. So when a pathogen like a virus enters a host, there are a number of defenses that get mobilized. Some of these, like antibodies, can be effective when the pathogen is in the extracellular space. But after a virus infects a cell and takes over its biosynthetic machinery, here the major host defense that is responsible for this is the CD8 T lymphocyte. It recognizes the cell as being virally infected and eliminates it. In this, we know the CD8 immune response is important for HIV/AIDS. And we know that based on a number of things, some of which are that if you look at the control of viremia after a patient gets infected, there's a correlation of the amount of viremia with the CD8 T cell response. And there is genetic linkages towards the evolution of the disease, and I'll talk some more about that. If one also looks at the evolution of the virus, the mutations that it has, you can also show that there's a selection pressure, and I'll talk about that. And this is also relevant because in HIV/AIDS, there are a number of associated viral comorbidities. Okay. So this is a little video that shows a CD8 T lymphocyte recognizing a virally infected cell. It makes a tight adhesion and it goes on and kills this cell, which then eliminates a little viral factory. So how is it that the CD8 T cell can actually tell that this is an infected cell? How does it recognize it? And what it's looking for is this structure here. This is an Mhc Class 1 molecule. All cells, virtually all cells in the body express this molecule. And CD8 T cells circulate looking for these. And if it finds one that it can recognize, then it will become activated and can eliminate the cell. So this is actually a T cell's view of the molecule. We're looking down on the top of this. And what the T cell is looking for is this structure here, which is an antigenic peptide. So the basic pathway I've illustrated here in this cartoon is that all cells express a subset of their genes and transcribe and translate those into proteins. All proteins in cells are turned over, and as an intermediate in that process, one generates oligopeptides. A fraction of those get transported into the endoplasmic reticulum where the Mhc Class 1 molecules form. So they will bind peptides of the correct length and sequence and take those out to the cell surface for display. So in this manner, cells express a sampling of everything that they've made, and this allows the CD8 T cells to look at these cells and see if these sequences are normal, in which case these cells are ignored. But if a cell is infected with a virus, some of the viral proteins will be made into peptides, and some of that information will come out on the cell surface that allows the CD8 T cell to see that this is a virally infected cell. Now these peptide binding receptors, these Mhc Class I molecules, are encoded in the major histocompatibility complex. It's on chromosome 6 in man. And this is the human MHC, the HLA locus. And there are 3 genes that encode these Mhc Class I molecules. These are known as HLA for the complex, A, B, and C. So we have 3 different Class I molecules that the system used to display all of this information about what the cell is making to the immune system. Now, if you think about this, a cell is making huge numbers of different proteins. If it's infected, it's expressing viral proteins. So there are going to be a very large number of different peptides that need to get displayed. Yet, we only have 3 different Class I molecules to accomplish this. So how does this actually work? And the answer to that comes down from the fact that these are peptide binding receptors. So again, this is 2 views of the same thing. This is the T cell view. The peptide here is bound in a cleft between 2 alpha helices sitting on a beta-pleated sheet. And when this was first recognized, this molecule bound peptides, that these receptors bound peptides, therein lay part of the answer for how a single molecule could bind so many structures. And so if you actually look at a peptide structure, this is a dipeptide, 2 amino acids linked via an amide bond. This would actually be too small to bind to a Class I molecule, but it allows me to make the point. So all peptides will have a free end terminus, a carboxy terminus, and then the structure of 2 carbons linked with an amide bond. And regardless of the amino acids, this is all common, what's shown here in green. The only thing that differs amino acid to amino acid is the side chain. So you could imagine that if you made a receptor that simply focused on the common regions of the peptide, you could bind all sequences, right? So to a first approximation, this is what the immune system attempted to do, but it couldn't get quite enough affinity to bind these peptides solely by focusing on the common areas. And so what it needed to do is to bind a couple of side chains. So this is a typical size of a peptide that binds to Class I molecules. And typically there are a couple of pockets that bind side chains, frequently the 2nd position and the 9th position. And there are only a couple of amino acids that would fit those particular pockets. But all of the other residues that don't need, that aren't binding into those pockets can vary between all 20 amino acids. And so that means that a single Class I molecule can bind a very large number of peptides, but not all, because it needs a couple of specific side chains here. So that would be true for the HLA-A molecule. The HLA-B, they're similar principles, but they're different pockets. Okay, so that can bind a very large number of peptides, but not all. Same thing with the HLA-C. And so via this sort of mechanism, a limited number of Class I molecules can bind a huge number of peptides, display much of what the cell is making, but not everything. Okay. Now, these Mhc Class I molecules, the HLA-A, B, and C, are the MHC molecules are the most polymorphic genes in our species. There are well more than 10,000 different alleles for the Class I molecules in humans. And so that means that my MHC molecules are likely different than yours, and yours are different than your neighbors, et cetera. Okay, so highly, highly polymorphic. So where do the polymorphic residues actually exist on the MHC class 1 molecule? And so we come back and look at the structure. This is the cartoon of the molecule sticking up, and this is the T cells view. The peptide would go down here. All of these residues that are colored in red are the ones that are very variable within our species. Okay. So that's going to change the molecular contours up here on the alpha helices and down in the groove. And so what is that going to affect? And what that's going to affect are things that are going to bind there, right? And so what are the things that bind there? One of them we just talked about, which is the peptide. And so it's going to influence what peptides the class 1 molecule can bind, mine different than yours, et cetera. And the other thing that's going to bind to this complex is the T cell receptor. And so it turns out, as you probably know, that T cells carry very much about the particular allelic form of the class 1 molecule. Now, the fact that this is so polymorphic and it influences these binding events has a number of implications clinically and also for things like vaccines. Because this means that different individuals are going to respond to different antigens, right? If my cells are displaying different peptides than yours, it's going to influence the specificity of the response and what we can respond to. And so this will influence potentially the course of disease and also a response to vaccines. And I'm going to illustrate that by an important study that was done now about 10 years ago that was looking at the genetic basis for individuals that are so-called elite controllers of HIV. So I think, as most of you know, there are individuals that get infected with HIV, which the majority of individuals will go untreated, will go on to develop disease. But there's a small subset of patients that clearly get infected and without therapy are able to control the virus, right? And so there was a lot of interest in trying to know what was unique about this set of patients that seemed to be able to control the virus. And so there was a very large GWAS study done looking at single nucleotide polymorphisms in a large number of patients. And remarkably, out of the study, so this shows all of the various chromosomes that were looked at, there was a single genetic locus that was highly statistically linked to this. And what this turned out to be were Mhc Class 1 alleles. So this subset of patients had a unique set of Mhc alleles, and this seemed to confer the ability of these individuals to control the infection. And the data that's emerging on how this actually works seems to be based on the things I'm going to be telling you about today. So in HIV, there are a number of potential epitopes that can be presented on Mhc Class 1, and many of these will go on and mutate to forms that no longer can bind Class 1. And that's evidence that the virus is applying a selection pressure. The immune system is applying a selection pressure on the virus, and the virus, since it is a very sloppy virus, is able to mutate these and escape detection. But it seems that there's an epitope, or there are epitopes present in the capsid protein that if it mutates, the virus becomes less fit. And so it's difficult for the virus to change this, because if it does, it doesn't any long, it's compromised. And so the elite controllers that have particular HLA alleles, HLA-B57, can bind and display this, and it's hard for the virus to escape this, okay? And based on this sort of analysis, the principal genetic determinants that allow these individuals to control the virus is Mhc Class 1. And the mechanism, we believe, is Mhc Class 1 antigen presentation. And this, I think, is among the evidence that shows really the importance of this. So the key points here, there are 3 different MHC class 1 molecules that are responsible for displaying peptides. They have a groove, a peptide binding groove that allows them to bind peptides. The peptides are bound via common characteristics of the amino acids plus some side chains. This permits these molecules each to bind and display a very large number of peptides. And the polymorphism within our species affects this process. It affects the binding. Okay. And this is important for Hiv. So this is a system that is binding and displaying peptides. Now your cells don't, when they synthesize proteins, do not make them as peptides. These have to be generated via proteolysis. And when the immune system evolved, which is really a phenomenon that evolved in vertebrates, it evolved these display mechanisms. Basically, the system looked around and saw that cells were already making peptides as part of the normal catabolism. So all proteins in the cytosol and nucleus are degraded, and that's via the ubiquitin proteasome system, into peptides, and those peptides are then broken down for things that are useful. The proteasome is a large particulate protease that's present in the cytosol and nucleus of all nucleated cells. And it's a large particulate protease. It looks like a cylinder. This has been cut in half. And you can see there's an aqueous channel that runs through this. And so proteins enter, they hit the active sites here, and are broken down into peptides. Now, when the immune system evolved and decided to use this as a source of looking at what a cell made, because everything the cell made got broken down into peptides, it also evolved mechanisms to modify this. And what it made was a distinct form of proteasome that we call Immuno Proteosome. So in this cartoon here of a proteasome, I told you the active sites are present in the center here. There are 3 different ones, each encoded by a different subunit. And it turns out that when the immune system evolved, it generated 3 alternate forms of these active sites, some of which are encoded in the MHC on chromosome 6. And these are constitutively expressed in cells like dendritic cells. And in all other cells of the body, these can be induced in the presence of inflammation, particularly in response to interferons, type 2 and also type 1. And when these get expressed and you start to synthesize proteasomes, these preferentially get incorporated, excluding these subunits. So you're changing where the scissors are, the active sites, and this makes a different set of cleavages, which makes a different set of peptides. And it turns out that these peptides, on average, are better for getting displayed on MHC class 1 than these, although these can get presented. These are better. And we know the system is important for responses. So key points here. In order to generate the peptides from the expressed genes, the immune system utilizes this normal system of protein degradation. The system also evolved these modifications of proteasomes to optimize the presentation of antigens. Okay. So we've gone now from having an expressed gene into a peptide, and now that peptide has to get to the class 1 molecule so it can bind and get displayed. So the class 1 molecules are transmembrane proteins, so these are co-translationally transported from ribosomes into the endoplasmic reticulum, where they fold assemble and fold into the structure I've shown you. And we'll come back to what's going on here, but I now want to talk about how they're going to get the peptides. So how is it that peptides can get into the endoplasmic reticulum? Because what I've told you is that these peptides are getting generated by the ubiquitin proteasome system, which I told you is present in the cytosol and nucleus. I also told you that the class 1 molecule is assembling in the center of the endoplasmic reticulum. So between these 2 structures is the membrane of the endoplasmic reticulum that's highly hydrophobic, And energetically, there's no way that a peptide of 8, 9, 10 residues can pass across this membrane. And so in order to get these two things together, the immune system had to evolve a peptide transporter that we call TAP. It's a heterodimeric protein that's its subunits are also encoded in the MHC. And this molecule, the heterodimer assembles like this. This is a picture of the actual structure. This was recently solved by cryo-EM. And what this is an energy-dependent transporter, so it needs to use ATP to get peptides across the membrane. It can transport peptides that are about 18 to 16 residues in length. The efficiency drops off very much after that. And it can transport a majority of the peptides that are made, but not all. So it's important for it to transport as many as possible because this allows the immune system to survey what a cell has made. But there's some limitations, and that makes some holes in what we can see. Okay, so coming back here, the class 1 molecule is generated in the lumen of the endoplasmic reticulum. The TAP transporter is allowed, it brings the peptides in for this to bind. And while the MHC molecule is in the endoplasmic reticulum awaiting a peptide, it associates with a number of chaperones and other proteins. And so this structure here is known as the peptide loading complex. And its structure was also solved by cryo-EM very recently. And so this is the cytosolic side, this is the TAP transporter. The class 1 molecule is here. And what I want to tell you about next is this molecule shown here, which is something called tapicin, okay? And there's actually a couple of related molecules that are important here. One is this molecule tapicin that's in the peptide loading complex. And there's a similar molecule called TAP-BPR that's outside of the peptide loading complex. And what both of these do, these are proteins that help load the class 1 molecule with peptides and edits the ones that they're going to be making. And this molecule tapicin also helps hold the class 1 molecule in the endoplasmic reticulum until it's loaded with a peptide, right? That's useful. You want these loaded with the optimal peptides before you send them out. And coming back to this editing function, the underlying mechanism for this has recently been elucidated. So here we are. This is, again, looking down on the top of an MHC class 1 molecule. These are the alpha helices here. You can see the beta-pleated sheets in the background. And if we have a class 1 molecule that binds a suboptimal peptide, so one that's fairly low affinity, which is not going to be so useful because that's simply going to fall off the molecule. What happens is that these peptide editors, as they're associating with the class 1 molecule, and you saw this picture of the cryo-EM and how intimate these are, that these editors have a loop that folds into the end of the class 1 molecule's groove. And so this is going to disrupt any peptide interactions with this end of the molecule. And so if you have a low affinity peptide here and you break some of the interactions here at this side, that's going to become even lower affinity, and these molecules are going to simply fall off. But if you have a high affinity peptide that comes in and binds fairly strongly, if you disrupt these interactions, it can still stay bound, right? And so this is a way that the system selects for higher affinity peptides. Okay, so the summary of the points here, that the peptides are generated in the cytosol. They are then transported by the TAP transporter into the endoplasmic reticulum. That's where the class 1 peptide binding receptors are forming, and they associate after they form with the TAP transporter and these chaperones. And these peptide editors allow the class 1 molecule to be held there till it binds a peptide and helps them load with the right sort of peptides. Okay. Now I want to come back to the peptides that bind to the class 1 molecule. It turns out when you look at these, they are all of an almost identical length. So in humans, most of these are either 9 residues in length or 10 residues in length. In the mouse, they're either 8 or 9. And the reason that there's such a uniform size is that the peptide binding groove is closed. So a long molecule is going to extend out, and that won't work. Moreover, the class 1 molecule binds the free end terminus of the peptide on one end and the carboxy terminus on the other end. And so that really fixes the length. If you can't bind there, it kills the affinity, and so the peptide won't stay bound. Okay. And so that then leads to the question, I've told you that the proteasome is responsible for generating the peptides, at least making some of the cleavages. Does it make these exact 8, 9, 10 residue peptides, or is there additional processing machinery that is involved in generating these peptides? So it turns out when this has been looked at that proteasomes frequently produce peptide precursors, ones that are extended on the end terminus. And so if this is a protein sequence and this is the regions that's going to bind class 1, the epitope, and if you look what the proteasome does, it's the only activity in cells that can make the proper C-terminal cleavage, but it often makes N-extended peptides. So these could potentially be used by the system if there was machinery that could take off these extra residues. And it turns out that such machinery exists, and that a major part of the trimming of these long peptides occurs in the endoplasmic reticulum. And the enzyme responsible for that has been defined, and it's something called ERAP1, or some people call it ERAP. And this is an aminopeptidase. That means this is a peptidase that takes off of the N-terminal side residue by residue. And humans actually have a second related one called ERAP2, so they have ERAP1 and ERAP2. So it turns out that ERAP1 is important. So if our lab and others have knocked this out from mice, and it turns out this markedly changes the peptides that are presented, at least in mice. And in humans, it's been found that there are polymorphisms, variants of ERAP1 in the population, that are associated with autoimmune diseases and also responses to other things. So it's an important enzyme. The system can still work without it, but not so well. Now, another aspect of ERAP1 that is very unique and very interesting is that it's able to trim peptides using a molecular ruler. And what I mean by this, if we take a peptide that's got extra N-terminal residues, so this is an 8 residue peptide, which would be a mouse epitope, that this enzyme is capable of trimming, taking off 11, 10, 9, but when it gets down to 9 or 8 residues, it slows or stops. And that number, 8 or 9 residues, should ring a bell in terms of what I've told you, because that's the exact size that an Mhc Class 1 molecule wants to bind. So this is really tailoring the peptides for presentation. So this is a very interesting activity, and there's been interest in trying to understand how this works. So the ERAP1 molecule has been crystallized and it's structured solved. It's shown here. The active site is found down here in the groove. And this molecule has a lot of flexibility. You see that it can open and close. Okay. And that's actually essential, we think, to this activity of the molecular ruler. Because what I'm going to show you next is a high-power view down here in the active site. And here we are. The active site is here. This is a zinc molecule. This is a zinc metalloproteinase. And when the molecule is open, as I showed you, it turns out that this amino acid here, this tyrosine, is oriented away from the active site. But when this molecule closes, this rotates and puts the tyrosine into the active site. And it turns out this is an important residue for the active site. It helps coordinate a water that attacks the peptide bond. And so what is thought, the way that this works, is that about 8 to 9 residues away from the active site, there's a trigger that triggers this molecule to close. Okay? And so the peptide is there, it triggers the closure, which is going to trigger the active site, make it active, and so the other side will get clipped. And that process continues until we get down to a peptide that's 8 to 9 residues, because now it's too short to hit the trigger and also have its fingers in the active site. And so it can no longer be trimmed. And that gives us peptides now of the exact size that are needed. Okay. So, summary of the points here in terms of generating the peptides that are going to be presented. The initial cleavages are made by the proteasome, and this often results, or most often results, in these long precursor peptides. The proteasome is making the correct C-terminal cleavage, and it's the only activity in cells that can do so. And other peptidases, like ERAP1, can trim the N-terminus of the peptides. Okay. So I think one of the reasons that it's important to understand this information is in terms of really understanding what the immune system is going to look at. So if you were to look at any particular protein, a viral protein, you can find many peptide sequences in it that potentially could be presented. Okay? They could bind class 1 molecules. But it turns out, if you look at what a cell actually makes, it's very few of those peptide sequences that get displayed. And that's a complex phenomenon. We know there are many mechanisms that contribute to that. But one of the ones that's important is whether those peptides actually get correctly cut out of the protein, because if they don't, they can't get displayed. And so this mechanism of generating the various peptides will influence the specificity of responses. And we're not there yet, but if we could understand all the specificity of all of these steps, we think our ability to predict what will actually get seen will get much better. Okay. So we've gone over the pathway here, how cells are capable of displaying a sampling of everything that they've made, and that allows the immune system to see, is this a good cell, or is this one that's potentially infected and that it has to get eliminated. Now, from the virus's point of view, the virus doesn't want to get eliminated. It wants to survive. It wants world domination. And so a number of viruses have co-evolved with the immune system and developed mechanisms to try to prevent their detection. And so many of these examples are seen in large DNA viruses because they have room in their genome to incorporate some of these extra things. And so I'm showing a sampling of these in this little cartoon. So our basic pathway here is that cells will produce an antigen that will get made into peptides that will go through the TAP transporter. The class 1 molecule gets formed in the endoplasmic reticulum. These guys get together with the help of the editors, and that information goes out to the cell surface. So they're in various herpes virus, such as CMV. They make a protein that when the class 1 molecule is forming in the endoplasmic reticulum, the CMV protein takes that and pushes it back into the cytosol, where it will get degraded. And so the cells are making the antigens, but there's no class 1 that can display it, right? So the immune system can't tell that. CMV and HSV make proteins that can block the TAP transporter. So you have the class 1 molecules, you have the antigens, you make the peptides, but these guys can't get together, and so we lose that information. And so there are a number of examples of these sorts of things. And it turns out that in HIV, although this is not a large DNA virus, it uses one of its multifunctional proteins, NEF, that can bind the class 1 molecule when it's on its way to going to the cell surface. And instead of allowing it to go to the cell surface, it directs it into the lysosomes of cells, causing the class 1 peptide complex to be degraded. Okay. How much time do I have? 10 minutes, okay. So what we've been talking about here is really the pathway that allows effector CD8 T cells to monitor the health of cells in the body, detect ones that are abnormal and eliminate it. But I'm going to tell you here about another pathway that's important for actually initiating the CD8 T cell response so that we can get those effectors. And that's something called the cross-presentation pathway. So the way that one initiates an immune response, this is actually shown here for cancer, but the same thing is true for infected cells, is that we have these abnormal cells present in the tissue. And the way that the adaptive immune system actually detects these is that it uses dendritic cells to be out in the tissues to look around and see if there's a problem. And if there is, to take information about that problem and take it into the lymph node where the CD8 T cells are to wake those cells up, wake up the specific cells that can then go out to find the cancer or virally infected cell and eliminate it. And it finds it via the mechanisms that we talked about. So the issue with dendritic cells having this job of having to collect this information is this raises the question of how can the dendritic cell sample antigens from a cancer cell or a tissue-tropic viral infection. And the reason that that's an issue is based on the mechanisms that I've told you about here. These antigens that are made by other cells can't enter the class 1 pathway because this is a pathway that's monitoring the cytosol and nucleus and what a cell is made. And these external antigens can't get into that pathway. They can enter the endocytic pathway, and that's important for generating peptides on Mhc Class 2, which I haven't talked about. So how are dendritic cells capable of gathering antigens from these kinds of pathogenic threats and displaying them on Class 1? And the answer to that is this pathway that's called cross-presentation. So what ends up happening in this pathway is that a dendritic cell that's out in the tissue and encounters a tumor cell or a virally infected cell will eat the antigens from these cells when they die, or there are other mechanisms that they can gather this. This will go into the endocytic compartment, which is important which is a compartment that's sampled by Class 2 molecules. But one of the other things that the dendritic cells can do is to transfer a sample of what they've eaten into the cytosol, which allows them to enter the Class 1 pathway that then allows the peptides from those to be displayed on Class 1. So when it gets to a lymph node, it can activate the CD8 T cells. So in terms of this process here, the pathway that's going on is that the dendritic cell will ingest the antigens from the cells in the tissues, transfer a portion of that to the cytosol that's now accessible to the proteasome that will make peptides, go via the TAP transporter to Class 1. So this is really the unique step that dendritic cells do. And there's been a lot of interest in understanding this pathway. And one of the evolving pictures here, and something that we're currently working on, is that if you look at the phagosomes that are involved in this, one of the things that ends up happening is that a number of molecules that are normally present in the endoplasmic reticulum, which I've told you in the first part of the lecture is really important for the Class 1 pathway, get transported and transferred to the phagosome via a couple of targeting mechanisms. And what this endows the phagosome with is machinery that will allow it to transport some of the antigen to the cytosol so this can get degraded by the proteasome into peptides. And these peptides can then go via TAP into the endoplasmic reticulum to the Class 1 molecule. But they also, it turns out that the phagosomes have gotten transferred with TAP so that the peptides can also be re-imported into the phagosome, and they also acquire Class 1 from the endoplasmic reticulum. So these become cross-presenting organelles, And it turns out we think that both of these pathways are important. And this is really a new one getting studied. Okay. So this will be my last slide, just to summarize what I've tried to tell you, which is that in stimulating responses from T cells, dendritic cells have to present the antigens that they've acquired. And if it's going to be a CD8 T cell, they have to present it on Mhc Class 1. And so they are endowed with this capacity to gather antigens that are in the tissues and present them on Class 1. That's a process called cross-presentation. And in order to cross-present, the antigens that are internalized via the major pathway, there's some other minor pathways that can occur, get transferred into the cytosol, which introduces them into the Class 1 pathway. And there's this data emerging that the phagosomes may actually become what we might call cross-presentosomes, or actually specialized vesicles for doing this. Okay, so I'm going to end here, and I'd be happy to take any questions.
Hello. Thank you, Professor, for a very nice talk. So we're a little bit delayed, but I think we have time for at least 3 questions. Remember, you guys have the app you can use if you guys are shy.
Thank you. That was just great. You showed us that CMV downregulates Class 1 MHC, and that would allow it to propagate. But do you have any thoughts as to why CMV is such an immunodominant molecule, despite the fact, or the virus, despite the fact that it limits its presentation?
Yeah. So I think that becomes sort of a philosophical question. There isn't actually data to say.
Brazil,
right. Filosofal. Yeah. So it's clear that these mechanisms do not completely eliminate recognition of the virus. And so responses are generated. And for some viruses, you can actually get pretty robust CD4 killer kinds of responses, which will not be affected by these mechanisms. Okay. But I think in the absence, so it's decreasing the ability of the virus to get detected, and that may be important in certain niches more than others, and may allow the sustained presence of the virus even when you're eliminating some of the cells. So I think it's a relative sort of battle.
Two questions. Is HLA-E presentation the same as A, B, and C? Number 1. And number 2 is where do the synectin and SEC22 come in cross-presentation?
Okay. So the first question, I can't answer how those compare. I simply don't know. So the second question that was asked was, what about SEC22B and its target molecule? And so what those are molecules that help vesicles that are generated in cells, which are going to transport various molecules here and there. How that allows them to fuse and get with their target site. So it's been shown that SEC22B is important for cross-presentation. And it's thought that it's helping some of these vesicles that come from the endoplasmic reticulum to fuse with the phagosome and deliver those. That doesn't explain actually how those vesicles target there, or how they're actually transported there. And work in our lab that's not published has identified a small GTPase, a Rab protein called Rab39A, that seems important for taking those, and it probably uses SEC22B for the fusion. And that's part of this maturing of the phagosome that I show where it gets the ER components.
Do we have more questions? No. So actually I have one. May I?
Por favor.
Okay, so you mentioned that some of the subunits of the proteasome, they are dependent on interferon, right? So interferon would definitely change the way the proteasomes work. So I was just wondering whether in different compartments of the body, for example, we have HIV in the intestines and we have HIV in the spleen. So has anyone ever checked whether what epitopes are being presented here or there, and whether, because intestine is more suppressive than the spleen and this kind of thing?
Okay. So in terms of HIV, lymphocytes, including T lymphocytes, are one of the kinds of cells that constitutively express the immunoproteosomes. So they don't need interferon for that to be turned down. So the expectation, and I don't know anybody that's looked at the data, is that in those various sites you would still get the same things presented. This is more of an issue in things like cancer recognition, where you may have cancer cells that don't have immunoproteosomes, where the CD8 T cells that are generated have been generated to immunoproteasome-generated peptides, and so you may have some mismatch, and that may cause some problems. But in the case of HIV, at least for T cell targets, there shouldn't be that mismatch.
Thank you. So thank you once again, Professor Kenneth, and thank you all for your attention. Have a nice lunch and see you guys later. Bye-bye.